so we are moving really fast. So we are now moving to the next one and the last one in this session. So that will be given by Dr. Josep Canals. He's from the University of Barcelona, and he's going to talk about the striatum and the development and mutant huntingtin, how can modify this development of the striatum. So, Josep Maria.
Okay, thank you very much, Silvia, for the introduction. First of all, I want to thank Silvia and Sandrine for inviting me and giving us the opportunity to show the results that we have developed in the last year. So the results that I am going to show here are unpublished results. So I think that these are this is the meeting to explain the things that are already going. So as you know, Huntington disease, it's caused by the, sorry. No. Huntington disease is caused by the degeneration of the GABAergic projecting neurons in the striatum, and also by associated degeneration of the cortical layers. Okay, but we are going to focus on the striatum this time. In the striatum is populated by 90% of MSNs, medium spinning neurons, that are striatal projecting neurons. These striatal projecting neurons can be subdivided, as most of you know, by two kinds of subpopulations: the D1, the DRD1 MSNs and the DRD2 MSNs, which generate the direct and the indirect pathway. The fine control of the stimulation of these two pathways allow us to control the movements, the specific movements of our body. However, when the D2 degenerate, there is a hypertrophy, there is a hyperkinetic movements known as chorea, and when the D1 degenerate, we can find the dyskinesias observed in the late stages of the disease. However, it is clear that this is a neurodegenerative disorder. The symptoms appear postnatally, perhaps very young at pediatric stages in some cases, but in most of the cases, they appear in the adulthood. But it is very strange that these symptoms appear very late, because the mutation is from the very beginning. So why these symptoms appear so late in the development? And for this reason, different groups, including mine, has been focused on the development studies in Huntington disease models. And as you heard, the group of Sandrine has been focused on that, and also the group of Ferdinando is focused on pediatric Huntington disease. And our group is very interested to characterize the molecular alterations that occur during striatal development at embryonic stages, but also to characterize the changes in the homeostasis of neural progenitors during this time. Just very briefly, you have to know that the striatum develops from the neurodevelopmental progenitors, and these progenitors localized at the wall of the ventricles can give rise to different kinds of telencephalic progenitors that will migrate to the mantle zone, where they are going to become neuroblasts. And following two different waves of neurogenesis, they are going to produce the DRD1 MSNs that also express GABA and substance P between the E12.5 and E13.5 in mouse. And the second wave of neurogenesis will generate the DRD2 MSNs, also that also express A2A between E14 and E15.5. So in our laboratory, we have focused on the study of the molecular changes, comparing wild type and one model of Huntington disease, that is the Z1Q175. And we have performed bulk RNA-seq at E16, just when the two waves of neurogenesis finish. And to better understand which are the changes that occurs during the progenitor stage and which are the changes that occurs in the mantle zone where we have the postmitotic neurons, we have dissected the two areas using laser capture. Okay? And we have compared all the samples generated. Samples from the germinal zone from wild type and Q175, and samples from the mantle zone from the control and mutant animal. Our results show that in the germinal zone, we can find very few differentially expressed genes. Okay? And this could be due to the large heterogeneity of progenitors that we have in this area. However, when we compare the mantle zone, we observe around 1,000 of differential expressed genes, and most of them were increased, were upregulated in the Huntington disease model. If we take a look on the genes that are upregulated, we can see that around 200 genes codified by no proteins, and 134 genes codified for different RNA biotypes. However, there is a large number of genes that still must to be confirmed experimentally. Okay? In the unregulated genes, most of the genes correspond to known proteins. And when we analyze the gene ontology, which are the main groups of genes that we find that the genes that are upregulated regulates the transcription of different complex, okay, and also affects the spliceosome and the RNA processing. We have also found that most of the upregulated genes regulates the cilium organization and cytoskeleton, and most interesting, they upregulate the synaptic and neuronal action potentials. So this could indicate that in Huntington's disease, there is a premature maturation of neurons during the embryonic stage. Keeping this in mind, we have seen that in the mantle zone, there is a downregulation also of the cell cycle factors or the cell cycle proteins, and there is a downregulation of neural development and neuron projections factors. So confirming that this could be due to an accelerated neurodevelopment during embryogenesis. Sorry. We have also found different metabolic pathways that are already described in adult models of Huntington's disease. To better examine these changes or this possible premature neurogenesis, we have performed different cell analyses, and the first thing that we have performed is the analysis of proliferation at E12, E14, and E16. We have analyzed the S phase and the M phase of the progenitors at these stages. And as you can see here, we have compared the wild type and the Huntington disease mouse models, and we observe that there is an increase of S phase at E12 and a decrease of M phase progenitors at cells at E12. However, at E16, we observe the opposite results, and no changes are observed at E14. This could be related to changes in the cell cycle, and these changes in cell cycle could alter the neurogenesis. So the next experiment that we perform is to analyze the birth dating of the cells in the striatum, and we observe that there is an increase of birth dating at E12. That, if you remember, it's when the first wave of neurogenesis occurs. And there is a tend to decrease at E14 and a decrease at E16. Keeping with these results, we observe that the retention of progenitors in the germinal zone is decreased in Huntington's disease. So this could be due to the accelerated neurogenesis. The pool of progenitors is decreased, and this exhausts the progenitors, and for the reason we observe a decrease in the birth dating in the later stages. However, when we count the total number of cells in the striatum, we did not observe any change at different embryonic stages. So this could indicate that perhaps specific subpopulations are affected in Huntington's disease. So we next start to explore the possibility that there is a differential regulation of the two pathways during embryogenesis. The analysis of the RNA-seq that we perform indicates that there is a different regulation of genes associated to DRD2 MSNs and DRD1 MSNs, as you can see, the opposite directions. To further examine that, we have used two reporter animals, one that expressed TD tomato under the control of the A2A promoter, and another that expressed TD tomato under the control of the DRD1 promoter. And we cross-mate these animals with our Huntington disease mouse model. And we observe that during embryogenesis, there is a specific affection of the neurogenesis of the A2A positive cells, the cells that drive the indirect pathway. And you can see that there is an increase in neurogenesis at E12 and E14. However, the DRD1 positive cells are not affected during these embryonic stages. This produced an increase in the number of A2A positive cells at E18. However, when we check the number of cells at P21, we were surprised by the decrease of total A2A positive cells at this stage. And the only explanation that can explain that is that there is a cell death at postnatal stages. We examined the cell death at different stages, and we observed no changes between E14 and E18. However, there is an increase in cell death when the natural occurring cell death period is done at P5. And this cell death specifically affects the A2A subpopulation. So, in conclusion, our results point that there is a different regulation of the homeostasis of the different subpopulations in the striatum. And we observe a premature differentiation of the due to A2A MSNs that generates cells that prematurely differentiate, and these cells are affected by the cell death at postnatal stages. Just to finish, let me to thank the collaborators that have been participated in this project, and mainly thanks to Cristina Vila, that is the student that we have in our lab that has done most of the experiments. And thank you for your attention. And if there is any question, I'm happy to respond.
Yes. So I will ask one question, because again, for the sake of time, although we have some time now. I was very surprised by your data on cell death, because, for example, in the cortex, we've been looking for cell death forever and we never found any cell death. So do you have any idea that could explain the difference? Or,
yeah, really not. We have not looked at the cortex, and so I don't know if there is more cell death or not in the cortex. It could be. My putative explanation, of course, the cortex is very broad. Perhaps the cell death is located in a specific area. I don't know if you have checked specific cortical areas to examine cell death. Because one thing that we have observed is that there is also a degree in the anterior-posterior striatum in all these phenomena. Okay, so perhaps the specific area of the striatum and the specific projections from the cortex could be affected as well.
Okay. And very shortly, so your data, so in your data, you would have a tendency to suggest that the developmental contribution could explain some of the cell type specificity later on in the susceptibility of these neurons, or would this be ideal?
Yeah, these are our hypotheses. However, with the results that we have right now, it's difficult to say. Okay. But the hypothesis is that, of course, we generate, or the patients or the models in Huntington disease generate neurons, but these neurons are not 100% well connected. And when the system gets stressed because of course, many different phenomena, then the cells start to degenerate.
Okay, thank you.
This is just our hypothesis.
Thank you. So in fact, I will close the session now. So thank you to Sylvia. Thank you for all the speakers. I think it was a great session. I have to say that I think it's very frustrating, of course, that we cannot directly interact and follow up on discussion. As the organizer told me right now, don't forget that if you want, you could ask questions on the chat, and the speaker will be available to answer these questions. And for most of them, for most of the speakers today, it's not very difficult to find their email if it's easier for you. So as a conclusion, to make it very short, I think, of course, all this talk show that we still need a lot of longitudinal studies. We still need to gain insight into the mechanism. And this altogether should allow us to integrate abnormal brain development in Huntington disease to our future therapeutic strategy. Sylvia, do you want to add something?
I think that we don't have time really, so
yeah, we have 1 minute.
And so just thanks to all the speakers, and see you next year in Italy.
Okay, thank you.